Well, this report from the Journal of the American Medical Association, published just in June of 2000, stated, based upon this knowledge of the deficiencies of detoxification, that Parkinson's disease may be triggered when individuals who have an inherited vulnerability to toxins in the environment are exposed to such agents. And inherited vulnerability is a key part of that finding. But we actually published a report back in 1997 in the Townsend Letter for Doctors in which I stated that individuals with specific genetic defects causing hepatic detoxification enzyme dysfunction may develop Parkinson's disease as a result of exposure to certain environmental xenobiotic chemicals proving neurotoxic. So I think it's really quite important that we look upon nervous system disease, neurologic disease, as not just being confined to the brain. As we've now explored, there's a very, very important interaction between what goes on in the liver in terms of detoxification and neurological problems. Further, it's important to recognize that these abnormalities of liver detoxification are not necessarily genetically determined in terms of disease manifestation, but perhaps there is some predisposition to disease. We know that it's certainly quite simple to test individuals now to determine their ability to detoxify, to evaluate their sulfation pathways, for example, by challenging them with acetaminophen and determining how they sulfate acetaminophen, and get results such as this, which demonstrate the sulfation pathway as well as others, including glutathione conjugation, which is also really quite important. When an individual does have sulfation deficiencies, I think it's important to consider that they may have elevated gut-derived toxins, and again looking at another system in the body, the gut, that they may have cofactor deficiencies, and most importantly perhaps that they may have ongoing xenobiotic exposure or toxic exposure, especially if phase 1 of this hepatic detoxification profile is elevated. We look at sulfate precursors because they are important in enhancing sulfation, and these include nutritional supplements, methionine, cysteine, taurine, and glutathione, and perhaps most importantly, N-acetylcysteine. So again, it's important to recognize that while there is this genetic component of these neurodegenerative diseases, it is not necessarily genetically predetermined that an individual will succumb to one of them. That influences in our environment have a profound impact in genetic expression. Well, now let's look at reactive oxygen species and recognize that indeed reactive oxygen species can also activate microglia. And to understand how this happens, we have to talk about this whole concept of excitotoxicity, or the energy-linked excitotoxic hypothesis. Now, in this model, we first look at a normal neuron in which ATP production by the mitochondria maintains a normal electrochemical gradient that we see in the lower right of the screen. When the electrochemical gradient is normal, a magnesium block exists in the NMDA receptor such that glutamate stimulation doesn't cause any real influx of calcium. In the scenario in which ATP production is compromised, in other words, mitochondrial failure or mitochondrial downregulation for any reason, then we have relaxation of this magnesium block in the NMDA receptor, and this allows the influx of calcium into the neuron, and what this does is enhances free radical production. It's the influx of calcium that is most responsible for this enhancement of free radical production. So we have a scenario in which mitochondrial dysfunction causes decreased ATP production, which then leads to partial neuronal depolarization and then persistent NMDA receptor activation by glutamate and, unfortunately, the persistence of influx of calcium. And perhaps more importantly, mitochondrial dysfunction, because of reactive oxygen species formation, leads to cytokine formation because of microglial activation. So I think you're getting the idea that what we need to focus on then, and really what is the foundation of these neurodegenerative conditions, is mitochondrial function. And there are a whole host of interactions with mitochondrial function that need to be considered. And these include genetic influences, drugs, xenobiotics, metabolic influences, and even viral influences. We talk about how the mitochondria protect itself. What are the mitochondrial antioxidant defenses that it uses to defend itself and therefore be able to maintain function? These include things like superoxide dismutase, glutathione, catalase, CoQ10. These are important antioxidant defenses. We know that the mitochondria are very susceptible to oxidative damage, and when this happens, energy production is compromised, and that further enhances reactive oxygen species formation. Well, let's look at this slide for a moment, which demonstrates life expectancy of various animals plotted against free radical or superoxide production. The relationship demonstrates that those animals producing lots of free radicals have the shortest life expectancy, and the contrary is true as well, which is to say those animals with long life expectancies are found to have the least amount of free radical production in their mitochondria. So it's important then to understand that life expectancy is related to oxidative stress, and it is inversely related to oxidative stress. So it's obviously of significant importance. So let's look at mitochondrial function and recognize that most of the activity in the mitochondria takes place at the level of the mitochondrial membrane. And what are the factors that are most important for maintenance of function of the mitochondrial membrane? Well, they are listed here: lipoic acid, glutathione, selenium, vitamin E, essential fatty acids, ginkgo biloba, and phosphatidylserine. But let's focus first on phosphatidylserine, recognizing that it enhances neuronal membrane stability and reduces cortisol production. But the important part for our discussion is, in fact, its role in membrane stability. This, therefore, would increase mitochondrial efficacy, mitochondrial functionality. Would it have any implication with respect to humans? It certainly does. In a 1991 Neurology article, it was found that supplemental phosphatidylserine in this 12-week study was found to actually improve the computerized and standardized neuropsychological performance of a group of individuals who were memory impaired, and they described it as a promising compound for the treatment of memory deficits that frequently develop in the later decades of adulthood. Other things that we consider with respect to the mitochondrial membrane, ginkgo biloba, certainly been studied for thousands of years. Well, how does it work? Ginkgo biloba, we know, actually provides very important antioxidant protection of mitochondrial membrane and actually reduces the consumption of glutathione as well as peroxide, rendering the mitochondria with less DNA damage. Would it be helpful? Well, this study shows that when you challenge rat cerebellar neuronal cells with a producer of free radicals, which in this case is iron plus peroxide, that if they are pre-treated with ginkgo biloba, the degree of DNA damage was significantly reduced. And of course, we're all familiar with the study appearing in the Journal of the American Medical Association by Dr. Labars, a fascinating report in which he treated individuals who were demented for a one-year period of time and found that 27% of the treated group actually improved on standardized cognitive testing. Again, looking at the mitochondrial membrane, we need to focus on vitamin E and recognize this report, again appearing in the New England Journal of Medicine, in which vitamin E was found to, in fact, be more efficacious than a so-called Alzheimer's drug, selegiline, in prolonging the time in which individuals who were treated could maintain their ability to maintain their own activities of daily living or stay out of a nursing home or even die. Focusing perhaps more on mitochondrial function and recognizing that the deficiencies of mitochondrial function lead to enhanced reactive oxygen species formation, let's first take a look at acetyl L-carnitine. Acetyl L-carnitine provides the very important job of shuttling long-chain fatty acids from the cytoplasm to the mitochondria, essentially bringing the fuel to the fire, and in addition has some membrane stabilizing effects as well, much as does phosphatidylserine. So in the presence of adequate amounts of acetyl L-carnitine, we see that ATP production is improved. This renders a repair of the electrochemical gradient, reestablishing the magnesium block in the NMDA receptor, and that would then prevent or preclude the influx of calcium and the reactive oxygen species that would have otherwise ensued. So the question then becomes, if acetyl L-carnitine is as powerful as we say in terms of mitochondrial function, should it be efficacious in the treatment of, for example, Alzheimer's disease? And in fact, this was studied by Dr. Thal back in 1996 and published in the journal Neurology, in which he provided individuals who had Alzheimer's disease acetyl L-carnitine and found that, in fact, the younger patients aged 65 years or younger did, in fact, decline more slowly. But clearly, one of the most important factors affecting mitochondrial function is coenzyme Q10. And indeed, this isn't really new information. We've known for many years that certain so-called mitochondropathies, disease